of degenerative symptoms such as our own body's stem cell decline and inability to get the right cues to repair and regenerate tissues, low-level increase in systemic inflammation. Ultimately, this really manifests as the aging that we all are experiencing and chronic age-related diseases. And so at Juvena, we leverage the most regenerative source out there, the secretome of stem cells. So these types of secreted proteins that are naturally enriched for their ability to develop every tissue in the body and regenerate and repair tissues in order to target those dysregulated pathways and restore health of tissues to more youthful, vibrant levels in order ultimately to rejuvenate tissues in order to rejuvenate lives. This has fueled a growing pipeline that we've been working on through our JuvNET AI platform that has fueled this pipeline over the last now 7 years and counting. And that includes a candidate that's actually now in clinical development in phase 1 that can enhance muscle regeneration. It's an insulin for muscle. It acts across multiple models of muscle wasting from aging through to genetic diseases. And we're first developing it towards a rare disease known as myotonic dystrophy type 1. We also have a lot of other exciting hits at various stages of discovery and preclinical development. And earlier this year, we announced a partnership with Eli Lilly in order to actually leverage our JuvNET platform for mining stem cell secretomes in order to identify lots of different proteins that can enhance muscle metabolism and improve body composition and weight loss quality in combination with the blockbuster incretin treatments that many folks are taking these days for managing their weight. Before I get into the way that our JuvNET platform works, just to kind of take a step back, I'd love to just share a little bit for those that might not be biologists or in the field right now of biomedical research or drug discovery, to talk a little bit more about how different artificial intelligence technologies are really revolutionizing the way we do research and the way that we discover and develop drugs. We're able to now use, and I would say actually it's gone to the point where any company who doesn't use AI is just behind, right? And so, even just a few years ago, when I first went out and tried to fundraise on Sand Hill in San Francisco with investors, saying that you're going to use AI for drug discovery was super exciting. But at this point, it's kind of like you're left behind if you're just not using this rapidly evolving toolkit of different types of machine learning, deep learning, and generative AI techniques for improving our ability to discover and develop drugs. And so that comes through multiple modes of support, from improving target identification, meaning the rapidly analyzing really large sequencing data sets. And there has been, as we all know, a revolution in sequencing technologies over the last decade that has led to just a transformative amount of now data that we can leverage for training models. It's also in even more recent years leveraging the latest in generative AI techniques for actually novel design of new compounds from small molecules through to proteins, very excitingly. And what we all promise, hopefully we'll start to see that on the market, is this accelerated timeline, right? Rapid drug discovery, the ability to generate new molecules and discover new targets, and generate hits in a way that is no longer bound by more traditional hypothesis-driven methods or mass amount of trial and error before you get there. There's also higher success rates when you leverage AI techniques in the various processes of in silico through to in vitro, in vivo, preclinical validation. And then what Juvena isn't about, but I know a lot of companies are really taking advantage of, are leveraging the latest in AI for improving the ability to repurpose drugs. There's so many drugs out there for certain diseases which might actually have immense potential in other indications, or who that have failed in the clinic for a certain indication, but we can actually take that shelved investigational drug and repurpose it for something new if we're able to better understand the biology and disease pathophysiology that it can target. There, of course, continues to be challenges, and I'm happy to share more during the Q&A about that. But with biology, actually generating wet lab biological data, unlike things like billions of data points that large language models, for example, are trained on, that is often a bottleneck, is that you have these great models, but there's just not enough data, high quality data, standardized data to really get the right type of training and outputs, right? I always like to say garbage in, garbage out. You know, high quality data in, high quality data out, and if the data set's too small, you also might be biased in the predictions being made. Some folks like to say there's a black box problem, where sometimes you get some exciting biology results, but if you can't really understand how the machine learning model, the deep learning model is actually working towards getting those results, it can make it really opaque and difficult to understand from a biological perspective. And you know, sometimes if you're too in silico, if you're too, like there are folks in the AI field that aren't biologists that say one day we'll just get away with wet lab research, and that's unfortunately just not the case. You always do still need that human in the loop. You need the ability to actually test, you know, those predictions in the wet lab, and so that will, you know, slow the process of AI-driven drug discovery, but it's an absolutely necessary step, that human in the loop and that wet lab validation. And, you know, for many of you to consider, you know, what careers you want to pursue as you know, go through college and grad school and get your internships, there is actually a major talent gap in those who really understand really that intersection between AI, machine learning, all the latest types of, you know, various classes of AI techniques and biology, and how to actually leverage that towards solving biological problems. So I think that will be, you know, a major need in the industry. And then, you know, there is a lot of course, upfront investment. A lot of these platforms, Juvena is really lucky that we built a platform that's literally now fueled a clinical stage pipeline. There's so many companies I know that have a brilliant ideas, but they just need 20 years to get there. And sometimes investors get really, they're not as patient when it comes to that. So for Juvena, we use AI, and you'll see in the coming slides, across various different cases. I'd say we like to integrate AI techniques into every step of the drug discovery development process, from in silico predictions and predicting what proteins and stem cell secretomes are active therapeutic, through to actually producing those proteins and testing them in cell models as well as in animal models, through to then taking those proteins and predicting better sequences and engineering them to actually have drug-like properties and translate them into drugs, protein-based drugs for people. And so to be, you know, more granular, we use machine learning models that decode into closed loop proteins, you know, as simple as even just machine learning classifiers that will actually predict proteins that are secreted from cells versus stay in cells, different types of deep learning models to predict the response of both cells and mice models to a drug and actually quantify it in a very unbiased way. We leverage different types of transformer models, large language models to infer protein sequences that enhance the stability and function of a protein that we discover to make that next gen, you know, version. And then I'd say that generative AI models to ultimately predict even novel sequences that improve functions, and really we see that as sort of the horizon in the next stage of drug development. Taking a step back, right, so let's talk a little bit about how does Juvena's platform really work for mapping the therapeutic potential of stem cell secreted proteins. Well, one of the first things we did in building our JuvNET screening platform was start with exciting data and novel findings that showed that we could actually take subfractions of proteins secreted by human stem cells, including cells like human embryonic or pluripotent stem cells, and as a cocktail, these thousands of proteins, when added into onto different either aged mice disease models or even human cells in a dish, were incredibly transformative in their ability to repair, regenerate, and target underlying mechanisms of aging to restore tissue health and function with huge potential. And what's not surprising is that proteins that are secreted from cells and that are either located outside of cells and tissues or circulating in blood are in fact the worker bees of our bodies, right? They are our own body's natural biochemical signals that will bind different cells and tissues in our bodies in order to instruct our cells to do everything from immune regulation, immune response to infection, to promoting different types of repair processes, from wound healing to organ regeneration. ultimately activating our own body stem cells that we have throughout our life in different organs, and regenerating tissue. So it's a really exciting class of proteins already with therapeutic proven potential, right? Think of drugs like, we all know, insulin, right, for diabetes, or human growth hormone. These are secreted proteins, but before JuvNET existed, these type of proteins were only discovered serendipitously or through very tedious, long processes like, fractionations of blood or urine, or, kind of trying to predict a needle in a haystack sort of mechanism. And, and in fact, there's actually so much that we still, even today, don't know about different types of secreted proteins. About 42% of proteins that are secreted have very few citations. 50, a lot even have like 0. We just don't know what they do. We have no idea where they're expressed, what tissues they are working in. And 30% of predicted secreted proteins have no known binding partners. So actually don't know what receptors they bind to, what complexes they form, and how they work. And so Juvena really saw that there was a major need of our ability to deconvolute an incredibly complex, potent class of proteins that can be turned into drugs to instruct our own bodies to repair and regenerate as we age for many different diseases. And so ultimately, the novelty of our platform is that we're creating this map of regenerative protein biology, linking different proteins secreted by stem cells to tissues in which they're exhibiting disease-reversing phenotypes, from enhancing muscle regeneration to improving metabolism and fat burning for improved weight loss for metabolic diseases, through to different treatments for fibrotic diseases. It's been really exciting, and we've made a ton of progress in the last few years. Where our bread and butter starts is really in the first step we took to leveraging the secretome of stem cells, which is by having developed a way through quantitative proteomics to build our own proprietary library of over 2,000 proteins that are enriched in secretomes of stem cells, such as human stem cell, human embryonic stem cells, mesenchymal stem cells, all sorts of regenerative human stem cells, compared to cells that lack those proteins that typically lack regenerative potential, right? Think of fibroblasts or senescent cells or just post-mitotic cells. You can literally compare, using quantitative mass spec-based proteomics, the difference of those secretomes, take those different cocktails of secretomes and compare them across different cell types to understand the phenotypes they're inducing. And that then allowed us to identify and build an in silico library of proteins that are enriched in all sorts of regenerative processes across their role in development, in metabolism, and regeneration. And then we were able to continue building that library by mining multiple different kinds of stem cells, from pluripotent ones that can generate every tissue in the body, down to lineage-specific stem cells that are only producing proteins that will generate, let's say, like just different organs specifically in the body. And that ultimately led to our library that includes all sorts of proteins that aren't just cytokines or chemokines, but enzymes, shed receptors, cell adhesion molecules, all sorts of classes of proteins that we can actually then leverage through our compounding database and map to identify factors that can influence all sorts of disease-modifying mechanisms. And then what's exciting about our AI predictive model that leverages this library to then predict phenotypes, disease-modifying effects, is it is compounding in nature, where as we screen, as you'll see in a moment, I'll walk through the screening steps, we're actually growing our database with more and more features of these proteins, from the type of phenotypes or no phenotypes that they're inducing in different cell models of diseases and animal models, through to biochemical features, evolutionary conservation features, etc., that actually enables us to leverage all those data points to retrain our models and become more predictive with every round of screening, and take all that information and apply it from one disease to the next through transfer learning type of mechanisms. And so the value of the library, the database, and the models itself is incredibly valuable, in addition to the biologics that we're actually now developing that came out of this platform screening. So now that JuvNET has an awesome over 2,000 protein library, we next built a robust 5-step screening process, our JuvNET screening platform, where we integrated AI into every step of the process to help us identify and predict proteins and then validate them for given age-related diseases. And so the way this works is on step 1, we take our in silico library and leverage our a combination of our AI-driven predictive model, as well as understanding and taking multi-omics data, and I'll talk about the different data sets in a moment, to actually rank order those proteins and predict which ones might be driving a specific phenotypic response, such as, for example, enhancing age or disease muscle regeneration. We then move on and actually produce those proteins recombinantly, and so this is where the wet lab comes in, and we test those proteins individually as well as in different combinations for their ability to mimic the original library in inducing that phenotypic response in an aged in vitro or disease model. Once we generate those hits, ideally human proteins and human cell models, sometimes we'll go right into mice if there's no good human in vitro cell model. But once we generate those hits in vitro, we'll actually take that information, the true positives, the negatives, the features that it generated, and feed that back into our models to retrain them in order to pull out more proteins that they're predicting have a phenotype. So with every round of screening, we're actually getting more iterative, more predictive in the process. At the end, once we feel we have sufficient hits to move forward, and that usually is at least 12 or more, we'll move on to in vivo target validation, where we're actually then testing these proteins in animal models of disease, whether it's natural aging, or obese, or diabetic mice, or a dystrophy. Once we show that we can inject this protein systemically in circulation as a secreted protein, and it's rejuvenating this particular disease phenotype, we then move on to take the best proteins that have freedom to operate, are manufacturable, are looking safe based on our databases, and we move on to do protein engineering and turn them into more drug-like molecules, and so that's what we call biologics. This has led to over 55 hits in 6 different organ systems. It's produced Juvena's entire pipeline and phenotypes across hundreds of different cells. And so ultimately, as noted, Juvena is actually integrating AI into every single step of this in silico, in vitro, and in vivo screening process. And so in the in silico, we are essentially learning and predicting what proteins in our library are inducing specific disease-modifying effects. During the hit screening stage, we are leveraging different types of deep learning models, for example, to actually quantify in a very unbiased, effective way, rather than having what used to be even just 10 years ago, right? RAs in the lab sitting there doing hand counting, believe it or not, right? We're really leveraging, of course, our statistical models and AI models to unbiased, rapidly quantify, and then we're leveraging the latest in different types of generative AI, transformer techniques, to improve our lead optimization, meaning taking a protein that's shown efficacy in a mice, a human protein, and engineering it for things like improved half-life, safety, bioavailability, etc. Ultimately, it's, AI is one thing, but we're leveraging AI models in combination with high-quality data sets, right? Data sets that will help train and help us improve predictions and quantification for drug discovery and development. And so that includes models and data around safety profiles of our drugs, around whether the proteins we're predicting are enriched in stem cell secretomes. We also look then at the proteins that they predict, and we'll analyze them for, do they change with aging naturally? Is this something that really leads to that loss of tissue homeostasis? As well as the disease context. Is there multi-omics data? Is there sequencing data? Is there proteomics data out there? Is there epigenetic, genetic, that really suggests that this protein is actually involved in the disease pathology, right? And that dysregulated pathways that lead to that loss of tissue health and the onset of degenerative disease. So here with sort of, I'll rapidly just go through at a high level examples of, us kind of getting into the weeds of some of these, models that we're using. There is one model, our in silico prediction model, which we've dubbed at JuvNET PQNet, which essentially is what we're using and leveraging a deep learning model that helps us take proteins from our library, predict them and rank order them for efficacy, and then we actually move forward and we'll screen them in patient-derived cells. So in the case of, for example, muscle, we're talking about aged muscle biopsies or dystrophic muscle biopsies. We then identify those candidates. We'll do iterative screening cycles. We assess for commercial potential and the safety mechanism of action to make sure that makes sense. We'll move on into our animal models and ultimately into manufacturing as noted. And an example could be one of the proteins that we engineered that forms the lead candidate that's now in clinical trials, which is human IGF-2. IGF-2 being insulin growth factor like 2, which actually drives muscle development during embryogenesis, the core driver of muscle development. And we found it's highly regenerative in aged people, hopefully, based on our in vitro models, and definitely in mice that are suffering from muscle wasting. The other thing that we like to do is when it comes to quantifying different readouts, leveraging high content imaging and deep learning, we'll do the other side. So what we'll do is, for example, this one, in like an animal, we'll leverage a deep learning model where we'll take our videos and our images and evaluate the distance traveled, the behavior of the mice in a way that's incredibly unbiased, that's leveraging our images, our video, and then the recognition, so that we truly also can truly trust those results and show in a very rapid manner when we're screening multiple compounds the ability of this drug to actually have functional benefit. In this case, it was distance traveled. And you can see just as a test, it really works well where you see young, healthy mice, 2-month-old mice are performing way better on average than older 3-year-old mice, which are like people in their 60s and 70s. Once we discover proteins and show that they work in animal models, we move to do protein engineering, which traditionally, the way that folks did this was not leveraging AI, but then to do really traditional protein biochemistry, things like fusions. You really look at a sequence and sort of guess, what kind of mutations you make to improve stability, to improve production, to when you inject it, to improve bioavailability, make sure that it's manufacturable in a very stable manner. Like often what we find is that's just as important as actually the protein that's perfect working is how can you actually make that into a molecule that you can make and distribute and use as a drug. And at Juvena, we started, right? When we started the company, we were starting with more traditional protein engineering, but with the recent advents in generative AI, in AlphaFold, it's just opened up so much opportunity in that next frontier of actually much more rapidly and effectively doing lead optimization and protein engineering. From our ability now to leverage these tools to improve lead sequence optimization, to taking an original sequence and improving it from its native structure, to strengthen it, to strengthen its binding targets, to lead target engagement, literally finding ways that we don't even understand. These models understand way better than an expert human protein engineer what type of changes will actually improve target binding, and oftentimes those changes aren't in the binding domain itself and other aspects of the protein. And then also de novo design, right? Like we can actually now feed sequences and just say, create a total new sequence that you think will be better than these sequences. And I think that's really the next generation of how we're building our library. Rather than it just being from natural proteins from stem cell secretomes, we're now adding, based on the base of all those secreted proteins, these new predicted proteins that are even more potent, more efficacious. And it's really an exciting time to be in the industry in the field. So with lead sequence optimization, ultimately we are leveraging different types of generative AI and transformer models for lead series mutations that are designed in order to help us basically, take the structure, predict actually a, modest changes to that structure. We'll actually produce them and then test them internally, and then as we test them internally, we can take the output of that testing in the lab, again in the wet lab, and feed that back into the model to do better at the next time around in terms of predicting, what sequences actually, based on the predictions, are working versus not working. We're also leveraging these techniques for de novo target binder design and target engagement screening, where we're essentially using these different types of diffusion models to essentially better understand, different types of sequences that bind a target of interest in order to ultimately create these new binding domains and improve the potency of proteins that way. And then, we're ultimately also fine-tuning these large language models to predict different types of domains and all-out just structures for novel drug discovery, basically, from creating completely new sequences outside of even just stem cell secreted proteins. So with that, I will take just the last few minutes to share a little bit more about how we've actually leveraged the platform for some of our, where we're at today, right? So one of the first candidates that came out of our platform that's now in the end, we're wrapping up phase 1 healthy volunteer studies for is JUV161, which is a fusion protein between a modified version of human IGF-2 and human serum albumin that acts to enhance muscle regeneration, improve muscle metabolism, muscle insulin sensitivity, and overall muscle survival in order to, over time, improve strength and function in multiple muscle wasting models multiple muscle wasting models, including those associated with aging, associated with diabetes, obesity, and even dystrophies. And so the way that we discovered this was first taking our first generation library, at the time was about just 1,000 proteins when we first created this library. We took in our PQNet model and rank ordered about 100 proteins that we predicted could drive aged or dystrophic human myogenesis, meaning new muscle fiber formation. We then went in vitro where we tested those proteins in human muscle precursors that were isolated from either aged individuals or individuals with different types of muscular dystrophies. And of those 100, we were able to discover 13 in particular that were actually targeting various aspects of muscle regeneration to restore the health of these cells to youthful levels and create new muscle fibers and nice fat fibers, just like what happens with young people when you go to the gym. But unfortunately, older folks, things start to go downhill. We then took those proteins and tested them in multiple models of muscle wasting, because ultimately our thesis at JuvNET is we want to identify proteins that are critical towards the tissue homeostasis and health of a certain tissue that often then manifests as its dysregulation across multiple diseases. And so by doing that, we can find something like what our first drug is, which is a true, this drug, insulin for muscle. It's something that's hopefully going to work for so many different kinds of diseases, from age-related through to dystrophies, from obesity, from metabolic conditions like diabetes, to help repair your muscle or regenerate it. And so there's huge, broad potential. And so we tested our candidates in 8 different models, and 7 here, but it actually turned out to be 8 now. And of those, about 8 of those 13 showed some form of efficacy in 2 or more of those models. But one in particular, JUV161, showed efficacy across all these disease models. And so for us, that was such a great signal that this is potentially going to be a blockbuster drug as our first drug to prove the platform and to develop it into the clinic. So we engineered it, and this is how it looks. It's, again, a modified form of IGF-2 fused to HSA that activates AKT and mTOR signaling and multiple other pathways to drive muscle regeneration, and it's acting to really restore AKT signaling because this core homeostatic pathway is downregulated and dysregulated in multiple disease models of muscle wasting. I don't show you, for the sake of time, all the data that we've collected, but we are so excited to now actually be wrapping up healthy volunteer studies for everyone is great, no problem so far. And hopefully next year we'll be getting data both in individuals who have a CAS limit mobilization, where over time, what, many of us have experienced, if you get an injury, if you have a cast, so rapidly, right? So rapidly you end up losing so much atrophy, and it actually takes a lot of time to build that back, and the older you get, the harder that gets. And so this will be a really acute model for us to quickly show whether our drug, just like in mice, is actually preventing that atrophy, and then we will go into people living with myotonic dystrophy type 1 as well. It'll be a very informative year for us. The platform has also been validated through the partnership that we struck with Lilly. That was about 2 years in the making, and ultimately we're leveraging our JuvNET platform to identify proteins that can enhance muscle metabolism, prevent muscle atrophy, and even induce hypertrophy in combination with the blockbuster weight loss drugs for either improved weight loss, or what's really important is improvement in body composition and weight loss quality. Ultimately, with many folks that are obese or just overweight and want to lose a little weight that go on these incretins, it's more severe the older you get, but through this starvation-induced mechanism, just caloric restriction-induced mechanism, and through some of what the actual incretin therapies are doing directly to muscle, you're getting weight loss, but it's not just fat. We will all wish it was just fat, right? It's fat and it's lean mass, might be some bone mass, right? And so the problem is, over time, that might, result in accelerated frailty, and the older you get, the worse that kind of gets. And so our goal is to improve that weight loss where you're targeting just fat and you're actually preserving muscle, maybe even improving muscle function, health, metabolism, to overall help restore your metabolism and have more durable and long-term weight loss for folks who are suffering or just experiencing obesity or any diseases associated with metabolic impairments. So this is the North Star of that collaboration. I won't read it for now, but it's a really exciting partnership that we kicked off. There was a lot of press around this. They invested equity in the company as well, and there's just a lot of hopefully future potential in the next few years. We'll see how that goes. Really exciting and nerve-wracking times right now for Juvena with the clinic and with kicking this off. I'll end by really thanking, of course, the team who were instrumental into being where we're at today. Juvena now has about 30 employees, as well as dozens of consultants. Every summer we love having interns, so if you're interested in that, come talk to me. And we have a full team that does all the wet lab work through to protein engineering at Juvena. And then in particular, I personally, as the CEO, just a messenger. It really has been incredible leadership when it comes to the JuvNET platform, the different types of AI technologies that we're leveraging. That was really driven, really in major part by my co-founder and our Chief Scientific Officer, Jeremy O'Connell, who also spent a little time at Stanford after his postdoc at Harvard, and he's a quantitative proteomics and machine learning expert. And then Dr. Tak Mai, having also, we all have that Stanford connection. He spent 6 years as a postdoc at Stanford. He's absolutely brilliant and just self-taught when it comes to deep learning, generative AI, machine learning across all types of AI, combined with drug discovery, and he is our founding employee and the senior director of our platform and discovery efforts. So with that, I would love to take questions. Aside from weight loss, yeah. Okay. We actually see potential. What's exciting about our platform is it's disease agnostic. We're ultimately building this database, and in fact, we have hits. I don't show it here in this presentation. We've generated hits across 6 different organ systems in multiple cell types where we have applications ranging from potentially treating things like liver fibrosis, osteoarthritis, pulmonary fibrosis, cardiovascular disease. We first really focused on muscle before moving to other metabolic organs, because ultimately our thesis was muscle loss is actually associated with all-cause mortality, and if we can find drugs that can enhance and repair muscle and improve muscle health, that will actually have effects across all aspects of healthy aging, in terms of improving cognitive function, improving cardiovascular function, lung function, everything. You know what I mean? We really believe it's that core, and there's just no drugs in the market, unfortunately. And then all of us, no matter how athletic you are, no matter how much you go to the gym, you hit your 30s, your 40s, and then you just have this accelerated year-on-year loss of muscle that it increases, with every decade, unfortunately. So, of course. Yeah.
Thank you so much for the talk. You mentioned that obtaining data is one of the challenges right now, and I guess that even now that we understand that multimodal data are even more important, to be an even higher challenge.
Yeah.
I'd love to hear a little bit more how you guys get to face this challenge, and how do you see the literature?
Yeah, that's a great question. We really quickly realized at Juvena the importance, like you said, of multimodal data. So we take multi-omics. Our basis is quantitative proteomics, because we're really looking at proteins and features of proteins and how to leverage that. But we also look at RNA sequencing, genetics, high content imaging, as well. And so I think it's the challenge, of course, is how do you combine those data in a way that can really be leveraged towards more thoughtfully creating predictions. And we, even at Juvena, are consistently limited by just the amount of data, whether it's the high content images, whether it is the, robotics obviously enabled everything, whether it's just the amount of omics data. So that is a challenge. Of course, I know that in certain fields or certain applications, you can kind of make synthetic data. But that's and I actually think, yes, we have even done some of that. We actually have done some sort of like synthetic omics and things like that to improve our training models. But that's how I think. I think just getting a lot of access, collaborating with other researchers, other institutions, really putting the money into the data generation, and then being creative with things like synthetic data can really, help solve some of these bottlenecks. Yeah.
Hi, just a very general question.
Yeah.
What is the biggest challenge of running Juvena so far?
The biggest challenge I think there is on an annual basis, depending on whether there's money in the bank or not in the bank. I'd say that the biggest challenge as a CEO always is in fundraising and keeping the ship afloat, the operations afloat, and be strategic with raising that. It's investor relations. And then as the company grows and the founders get more and more diluted, it's managing the board for me personally. Yeah.
The human body is highly important, and how do we reconcile fixing one individual tissue when the rest of the organ remain aged? For example, Kamala had 20 years old muscle, but 70 years apart. And I guess my question is, what is the current approach to balancing tissue level rejuvenation, regeneration versus the whole systematic health?
Yeah, I think those are really, really good questions. To answer your first, can you have a 20-year-old, muscle and an 80-year-old heart? In fact, the rate of aging for everybody actually does vary in that manner. And one of the labs that I did my postdoc with, Professor Tony West-Cray here at Stanford, he actually really studies that by looking at these multi-omics approaches and even machine learning models to predict and actually understand which organ in your body is driving aging or more age than others, And that often leads to some people being, more susceptible to cardiovascular disease, to neurodegeneration, to, different kinds of things that affect us differently. And, ultimately, what I'm interested in is finding systemic proteins where they might work in one tissue, but we're actually looking to make sure we understand if they're actually working in other tissues and if they could more holistically improve communication between organs and multi-organ health. So it's critical that we get there, and that's also a very unique and different way than the traditional way that Big Pharma looks at diseases. They usually look just at one system, targeting one, without really realizing that actually sometimes these pleiotropic effects can be really positive for restoring synergies and homeostatic mechanisms. I don't think I remember your second question, sorry.
Actually, I wonder, can AI play a role in bridging this gap?
Absolutely.
Also, advances, for example, in vitro, animal, organ, the whole body.
Yeah, and I think, and there are a lot of companies out there now that are doing things like, different sensors that are taking constant inputs from your, from just your day-to-day, your actions, that are then feeding into these models that are, trying to create this almost like map of your health. So I know that there is a lot of progress there, and, great insights, great ideas too, if you want to start a company. Yeah.
Yeah, thank you very much for this great talk. I
Thanks.
just had a quick question, just out of curiosity. You developed this drug to help with muscle atrophy and recovery and stuff like that, but is it also possible, like you said, it's also can be used for hypertrophy? So also, is it possible to use this drug in like a performance enhancing matter for athletes or as a steroid?
Yeah, we get that question a lot. I'd say that the way that we're leveraging JUV161, our lead, is that we're taking something that's like this in young people, that becomes like this in disease or in age, and we're just restoring it to here. So when we actually test the dose we use in young animals, in very few metrics, is it acting like a steroid or boosting from just young, healthy baseline? But it might be different for people. Of course, we have yet to see that. But there are obviously a lot of drugs out there that really do, just, you just gotta be really careful, because it's more about restoring the health so that you're mimicking a youthful environment versus whenever you overdo anything, something ends up giving. And, that's why you have all these side effects from steroids. Alrighty, well, this was a ton of fun, and obviously if you have more questions, if you want to get in touch, my email's here in the thank you, and I appreciate your time and wish you guys the best of luck.